G-protein coupled receptor adaptation. So when we're talking about G-protein coupled receptor adaptation, we're meaning a type of adaptation that occurs at the cell membrane where the amount of active receptor is either increased or decreased. We talked about this previously. If I have two membranes, an equal amount of signaling molecule extracellular for both, but on one membrane I have, let's say, 4 receptors that are active, and another membrane I only have 2. Well, just by the rules of the Ka, because this is receptor plus ligand, giving me receptor ligand, and Ka equals to concentration of receptor ligand over concentration of receptor times concentration of ligand. If the amount of ligand is the same and I increase or decrease the amount of the receptor, well, the amount of the receptor ligand will also increase and decrease with how much receptor I have. So one way is to just increase or decrease the number of the receptors on the membrane. But another way is that the number of receptors on the membrane doesn't change, but there's some sort of negative feedback, decreasing the number of active receptors on the membrane. And it has the same effect. It's like the receptors are not there. Okay? Negative feedback. In every signaling pathway, there's a negative feedback mechanism which is itself activated by the pathway. And this is important because for some responses, you don't want the response to persist even though the signal is still there. So if we have a signal coming into the cell that is persistent, that is not going away, and we have activation of a receptor, which leads to an activation of a signal transduction pathway, which leads to a specific response, within that response, there could be included a negative feedback on the receptor. So as the signal persists, the negative feedback also persists, and there'll be a deactivation of the receptor so that this does not occur as much anymore. So the response starts to wane. And as the response starts to wane, the negative feedback is lifted. The example that I use often is inflammation. Inflammation is a good thing, but if it persists for a long time, it can be a bad thing. So there's a negative feedback loop on inflammation so that after a while, it is turned off, because it can lead to bigger problems beyond tissue damage or the disease that caused the initial signal. Therefore, the higher the incoming signal and the activation of the signal transduction pathway, the faster the rate by which the negative feedback builds, because the negative feedback is dependent on the signaling pathway being activated. So the more the signaling pathway is activated, the more the negative feedback builds up. Since the negative feedback mechanism is dependent on the pathway activation, the repression of the pathway will lead to the negative feedback being removed. So once the negative feedback is working and repressing the pathway, then what will happen is that the negative feedback will be removed and the pathway goes back to its normal state. So we're going to talk about one way that this happens, at least as far as the receptor is concerned. Desensitization of G-protein coupled receptors. Remember, desensitization doesn't mean that you're actually working on a single molecule. We're talking about this right here. The membrane is more sensitive to the signaling molecule here because there are more receptors available for it to interact with, where it's less sensitive here because there's less receptors for it to bind to. An activation of a G-protein coupled receptor will lead to a decreased sensitivity to signaling molecule. This is a form of adaptation. G-protein coupled receptors can be activated through the phosphorylation of their intracellular domain by pKa, PKC, and G-protein coupled receptor kinases, GRKs. GRKs are activated by G-protein coupled receptor pathways. So somewhere in this signal pathway with G-protein coupled receptor pathways, what you're going to have as part of the response is activation of GRKs, or a GRK. And this will go back to inhibit the receptor. Okay? Also, pKa and PKC are capable of phosphorylating the intracellular domain of their G protein-coupled receptors. A phosphorylated intracellular domain is a ligand for a class of proteins called arrestins. Arrestins will bind with high affinity to the phosphorylated intracellular domain of G protein-coupled receptors and block receptor interaction with its trimeric G protein. That is what is being shown here. In this case, it is a GRK that is responsible for phosphorylating the intracellular domain of a G protein-coupled receptor. And once that intracellular domain of the G protein-coupled receptor is phosphorylated, it becomes a ligand for this protein called arrestin. Arrestin will bind, and when it binds, it will not allow the interaction of the G protein-coupled receptor with its trimeric G protein. It simply is a physical block. This inhibition is removed by phosphatases, which dephosphorylate the G protein-coupled receptor intracellular domain. So this is kind of always active. So how would we draw this out? It's a bit more difficult to draw this out because you have this other protein called arrestins, which are involved. So when we're writing this part right here, this inhibition, we're not putting in the fact that GRKs are not directly inhibiting the receptor. They're phosphorylating the receptor, the intracellular domain of the G protein-coupled receptor. And let's just go ahead and make this into G protein-coupled receptor. And that leads to arrestins binding to the G protein-coupled receptor. And this could also be pKa or pKc, right? But this negative feedback is being reversed by phosphatases. These are specific phosphatases that their substrate is phosphorylated G protein-coupled receptors. So you can think about it this way. Where here it is inactive because it will bind to arrestins, and here it's active because arrestins can no longer bind to them. Okay? So if the negative feedback is lifted, for example, this is gone, well then the G protein-coupled receptor will be dephosphorylated, and then that will lead to the further activation of the signal transduction pathway if the signal is still there, right? And this will lead to the response, but this will also then again lead to these proteins being activated and leading to an activation of the G protein-coupled receptors through phosphorylation of the intracellular domain and binding of arrestin. Negative feedback through gap activity. G protein-coupled receptor pathway activation also leads to the activation of gaps that are specific for the G protein in the pathway. An activated gap will then substantially increase the rate by which a G protein hydrolyzes its GTP to GDP and thus inactivating it. Just remembering what gap activity does, this was our picture from before, and here's our gap. And remembering that a monomeric GTPase, which a G-alpha subunit is a monomeric GTPase, or GTP binding protein, that it has GTPase activity. So the gap actually is a reversible activator of the GTPase. And this will lead to increasing the rate by which the GTP is hydrolyzed to GDP. We never said how the gap is activated. Well, the gap is activated by the signal transduction pathway. So when we're looking at the signal transduction pathway and we have signaling molecule activating the G protein-coupled receptor, which activates the G protein, and this will eventually lead to other things being activated and will lead to a response. Well, as part of that response, or somewhere in the pathway, you will get activation of gaps. And the gaps will go on to inhibit the G protein. That is through activating further the GTPase activity of the G-alpha subunit, thus leading to it taking the GDP that it's bound and hydrolyzing it to GDP, sending it to the inactive state. An activated gap will then substantially increase the rate by which a G protein hydrolyzes its GTP to GDP and thus inactivating it. This leads to a decrease in concentration of activated G-alpha and G-beta gamma. Because once a G-alpha subunit has hydrolyzed a GTP to GDP and it's inactive, it will go on to bind the beta gamma subunit, and the beta gamma subunit becomes inactive when it's bound to the alpha subunit. And you get the formation of the trimeric G protein again. An example is the regulator of G protein signaling RGS, which is a gap and is activated by G alpha. So in this case, the activation of G alpha leads to the activation of the regulator of G protein signaling RGS, which itself goes on to inactivate the G alpha by increasing the rate by which the G alpha hydrolyzes the GTP to a GDP. So if we're going to write it out, we can go something like this. We have signaling molecule activating G protein coupled receptor. This leads to the activation of the G protein, but we're just going to put G alpha here for now, okay? And this goes on to activate other things, which eventually leads to the response, but the G alpha also activates RGS, and this will go on to put an inhibition on the G alpha subunit, and that inhibition is in the form of the RGS binding to the G alpha subunit and increasing the rate by which the alpha subunit hydrolyzes its GTP to GDP, right? Now, the question that could come up is, well, this is a weird type of negative feedback because you're sort of shutting off the signal transduction pathway immediately. And that is not necessarily the case because there's going to be a delay where this happens. Okay? Now, the delay could be long or short. That is all to do with how much of this RGS there is in the cell, where this RGS is in the cell. Remember, the G alpha subunit is stuck on the membrane, right? So depending on all those factors, there could be a long or short delay before this happens. There will be a response, but you will also have the shutting down of the pathway if the signal is persistent. And if this negative feedback does happen, right, which it will happen, then once you have this sort of deactivation of the G alpha subunit, well, then that leads to less of this activated RGS. So this will make this negative feedback go away, and if the signaling molecule is still there, then you're going to have more G alpha or G protein activation, which will then lead to, again, having that negative feedback be activated. So it just depends on whether that delay is short or long, and you'll get different types of profiles as far as the response is concerned. And just to kind of see what that will look like, if we have our y-axis be the response, the intensity of the response, okay? And our x-axis is going to be time. And time could be in minutes here. And this is where we put the signaling molecule in, okay? So this is the part where we introduce the signaling molecule. And we'll have a maximum right here so that I don't go above it accidentally. If we say that we have a long delay before the negative feedback starts to take shape, okay, then what it'll look like is that you have no response, and then you have the response at maximum, but then the negative feedback takes over and it goes down. But as the negative feedback goes down and the response decreases, well, the negative feedback is dependent on the response. So now, what you'll have is, again, an increase in the response, but as the response goes up, the negative feedback increases. And you have something like this. And this is important to biology, because some responses have to be in this shape, isolating between maximum and going back down, and then going back up, and then going back down, okay? Now, if we have a short delay instead, the same thing is happening, but it's happening so fast that it looks something like this. You have that initial spike in the response, and then it'll go down, and it'll basically sort of stay at a lower level as long as the signal is there. Sometimes it could be very dramatic, like it could be something like this, where you have this maximum response, and then it goes down, and it just sort of stays right here because the negative feedback is being turned on and off very quickly. And this is important to biology because some responses require an immediate spike in the response, and then the intensity of the response is lowered to achieve the correct biology. Okay, we'll stop there and continue later. Thank you.